everyone, and welcome to BEaTS Research Radio, a podcast and YouTube channel dedicated to communicating science to the community. My name is Nicole Chu, and I'll be your host on today's episode with our very special guest, Dr. Carole Yauk. Carole holds the Tier 1 Canada Research Chair in Genomics and the Environment and is a full professor in the Deptartment of Biology at the University of Ottawa. Her research lab, called the GREAT Lab, is focused on the development and implementation of genomic approaches to measure how toxic chemicals can alter the function of genes. Prior to her role at the University of Ottawa, Carole led the Genomics Laboratory in the Environmental Health Science and Research Bureau for 20 years and was an Adjunct Professor of Biology at Carleton University since 2006. She is also the president of the Environmental Mutagenesis and Genomics Society. Thanks for joining us today, Carole.
Thank you for the invitation. It's a great pleasure to be here.
We're so happy to have you on BEaTS Research Radio. So as I mentioned before, you are the principal investigator for the GREAT Lab. And first off, the GREAT Lab is such a creative and innovative name. So could you tell us more about what this GREAT Lab stands for?
For sure. When we started the lab, almost a year ago now that I joined the University of Ottawa, we knew we had to have a good nickname for the lab. And so we went with GREAT because we wanted to be a great lab. And GREAT stands for Genomics in Regulatory and Applied Toxicology. So G for Genomics, RE for Regulatory, and Applied Toxicology is the important addition there. So genomics is the study of all the genes and genomes, the structure and function in the organism that you're studying. And so it broadly encompasses all the genetic material in an organism across that sort of more global landscape, as opposed to genetics, which looks at genes, more sort of individually.
Oh, that's great to hear. And could you talk more about the research program currently at your laboratory?
Yeah, we have two main areas of research in my lab. One area we call toxicogenomics. So toxicogenomics is combining studies in genomics with toxicology. It's really trying to understand how toxic chemicals change how genes are regulated and the output of the genes and how that's associated with disease. The other area of research in my lab is environmental mutagenesis, which we can also call genetic toxicology. And what we do with environmental mutagenesis more specifically is we try to understand how environmental chemicals or your exposures cause damage to DNA that can lead to mutations. So even within that subdiscipline, we dig a little bit deeper. So we distinguish somatic cells from germ cells. So germ cells are sperm and egg, which are passed on obviously to your offspring. So that DNA would be passed on to your offspring. So we're looking at heritable genetic effects in that case. So the DNA damage can lead to mutations that you pass on to your offspring if they occur in germ cells. Whereas the mutation work that we do in somatic cells are any other cell in your body are somatic cells. And those kinds of mutations can lead to diseases like cancer. So those are the sort of main areas of research in the lab. And it's really focused on trying to understand how environmental chemicals can cause toxic effects and focused on developing new tools to study and test whether chemicals cause these effects, focused primarily on human health risk assessment.
Oh, so you've mentioned the environmental chemicals. What are some sort of examples of environmental factors?
So we're living in a soup of chemicals, every day in our food, in our air, in the consumer products that we use. We in genetic toxicology, we focused a lot on combustion byproducts known as polycyclic aromatic hydrocarbons, and these are present in tobacco smoke and air pollution, and also. We get a lot of that through our diet. So when you're grilling your food and barbecuing your food. So these chemicals can interact with DNA. And when they bind to DNA, when the DNA is replicated, mutations can arise or breaks in the DNA can arise. Other areas we work in are in the bisphenols area. So you've heard of bisphenol A. There's hundreds of substitutes now for these bisphenols, and we're trying to understand their toxicity. And another area that we're working in is in a big chemical group called PFAS, per and polyfluoroalkylated substances. In the news, you might have heard of PFOS and PFOA. So if you watched the Mark Ruffalo movie, Dark Waters, or you saw the documentary, The Devil We Know, that's about these PFAS chemicals like PFOS and PFOA. And there's actually over 4,000 of them in our environment. So these are some of the chemicals that we study in our environment today in my lab.
Wow, that's definitely a different multi-array of chemicals that you guys are studying. So can you tell us a bit more about the main motivation behind your lab work?
We're really aiming to address a big problem in regulatory toxicology testing. And that's that, we've for decades, regulatory agencies have relied on tests in animals to predict whether there are going to be human health consequences to a particular chemical, to exposure to a particular chemical. And the problem with this is that these tests are very inefficient. They cost a lot of money and they use a lot of animals. And so it's very slow going chemical by chemical using these sort of rodent-based tests. And I mean, by today's standards, we have better tools available to us. And so our lab is motivated to apply these better tools, like tools in genomics, to help increase the efficiency of regulatory testing. So let me give you an example. If you want, if a regulatory agency is interested in whether a chemical may cause a carcinogenic risk, they may want to do the standard regulatory test that's used, which is the rodent 2-year cancer bioassay, which takes about 3 years to do and costs several $1 million and uses about 900 rodents. So, that it's just impossible with the tens of thousands of chemicals in our environment to actually have tested all our chemicals. So as a result, thousands of chemicals in our environment have never been tested. So we're developing higher throughput methods that look across the genome, so more broadly instead of one endpoint at a time, to tell us what are the mechanisms that are changing as a result of chemical exposures, and can we use that to more rapidly and efficiently and accurately predict potential human health consequences. So that's really the motivation, is to modernize how regulatory testing is conducted. And we're doing this kind of one test at a time, but the test covers a broad scale of biology, and we can do it much faster and apply it in a much more parallel way with other tests.
Oh, that's so interesting to hear. Thanks for sharing, Carole. And what are some of these applications to public health? You've mentioned the environmental mutagenesis in the study of cancer. And is there anything else as well?
We're focused really on chemical screening in our toxicogenomic program. So that's to identify priority chemicals that we should study in more detail. So it's kind of we apply a tiered testing approach. So we use cell culture models and we screen them broadly across a lot of biology. And if there's not a lot going on, then those chemicals become lower priorities. And if there's a lot going on, then those become higher priorities to direct future research programs to study those chemicals in more detail. In genetic toxicology, what we're really focused on is developing new tools to more deeply understand how chemicals cause genetic effects. And with genomics, we can look at any of the genes in the genome, and we can look at any species, whereas our current regulatory tests use, standardized animals and models like bacteria. So we really want to take it a step further and be able to understand in humans who are exposed to mutagens, what are the potential consequences to their somatic cells and their germ cells. So there's a lot of public health implications to the methods that we're developing. Right now, it's a lot of, as I said, methods development and not so much application in humans in the genetic toxicology area, but we're moving in that direction.
Yeah, we'll definitely keep our eyes peeled for the new innovative work that's coming out of the Great Lab. So do you mind going through with us some of the current research efforts underway in your lab? You've mentioned the toxicological methods, and could you elaborate more on that, please?
Sure. Well, let me give you a couple of examples. So I mentioned PFAS. So one of the things that we're doing with PFAS right now is we're studying liver organoids, which are like little mini human livers in a dish. And these liver organoids are composed of liver cells from 10 human donors. So they encompass a broader sort of set of the population, 5 males and 5 females. And we look at these little organoids and we expose them to PFAS. So we use those prototypes like PFOS and PFOA. But then for the PFAS that have not been studied, we're asking, how do these liver organoids respond to those new PFAS, and how does that compare to how they respond to PFOS and PFOA? And that helps us to indicate, to determine which PFAS should be priorities for risk management purposes. So we're using genomic tools to, in a really high throughput way, screen PFAS chemicals in the liver organoids. We're doing something similar with breast cancer cell lines and bisphenols. So bisphenol A is our prototype in this case, and we're looking at dozens of bisphenols now to determine, are they as potent as bisphenol A in these cell lines in their ability to cause toxicity to these cells. In genetic toxicology, we're really excited about a new sequencing technology, DNA sequencing technology that we're working on, which is called error-corrected sequencing. And this technology allows us for the first time to accurately determine mutation frequencies and rates at single cell level. So being able to really nicely characterize and precisely and accurately characterize mutation rates in animal models that we're using and in cell culture models that we're using. So we're developing this technology and figuring out how we can use it in regulatory toxicology testing.
Wow, that's so cool to hear. And do you have any important discoveries or advances that you'd like to share apart from that error sequencing that we just went through?
In terms of recent discoveries, really what we're doing right now is innovating how to analyze these big data sets. So the biggest challenge with genomics is that we produce a massive amount of data in a single experiment, and it can take a very long time to analyze those data if you do it in sort of a more conventional way. So we're developing computer toxicology, computational toxicology methods, and bioinformatics is the discipline that's paired with biostatistics to help us figure out the best ways to model the data, to understand how to put this information together to make decisions about the potential risks that chemicals cause. So we're developing a lot of tools that we think are pretty innovative and we're excited about to help us in that area. In genetic toxicology, one of the things that we're doing right now is we're focused on understanding whether exposure to these PAHs, the polycyclic aromatic hydrocarbons that I mentioned, cause mutations in sperm that can be transmitted to the offspring. So there's never been a study that's ever been able to definitively show that chemicals cause mutations in the male germline that are transmitted to the offspring. And the evidence space is growing now, but we're conducting studies on males who smoke and males who don't smoke, and looking at their whole genome sequence compared to their offspring to see if they're transmitting more mutations to their offspring. So we're discovering all kinds of very cool things about age-related effects in males as they age, and increases in mutation rates in their sperm. So stay tuned to see if we can actually, for the first time, discover a male germ cell mutagen that's causing heritable genetic effects.
Wow, that's really interesting to hear about the heritable genetic effects, and also to see how quickly your field is advancing. So for our last final question, a more open-ended question, in the next 30 years, what major challenges or obstacles in your field would you like to see solved or overcome?
So there's a major challenge globally, and that is to reduce or eliminate animal testing. For example, the US Environmental Protection Agency has a mission that by the year 2035, they're going to eliminate animal testing from their program. And this is a very important goal, and it's globally supported. There's industries like the cosmetic industry in the EU that have already moved away from animal testing. The major challenge is finding those cell culture models and the computer models that we can use to accurately predict health effects. So that's going to be a major focus of our work and the work of many people around the world, is ensuring that we have the right tools in place to be able to eliminate the animal tests entirely and feel confident that we're adequately protecting human health. In the field of genetic toxicology, as I mentioned, we have these incredibly powerful new tools that allow us to move away from needing to do mutagenicity tests in, for example, transgenic rodents or special model organisms that have, reporter genes in them that we use for genetic toxicology. Instead, we can now sequence genomes. And so the challenge is for us to figure out the best way to do that and to be able to begin to implement that in human health and environmental assessment. I envision a time in the future when we can all know our baseline mutation rates, for example, our baseline mutation frequencies and our potential baseline mutation rates. So these are areas where there's going to be a lot of work in the upcoming decades that we're excited to be part of that momentum.
Yeah, that's great to hear. Thank you so much for sharing on that. And thank you so much, Carole, for joining us today and for speaking on the innovative work at the Great Lab.
It's been a great pleasure. Thank you very much.
Yeah, we were so happy and honored to have you on our channel. And thank you to all BEaTS listeners for your time and for taking a listen to this wonderful episode. Don't forget to like and subscribe to our YouTube, Twitter, and Instagram channels at BEaTS Research. BEaTS Research Radio is supported by the University of Ottawa, the BEaTS Research Lab, and the Department of Biochemistry, Immunology, and Microbiology at the University of Ottawa. We hope to see you all again next week. Thank you.